present the EpiEndo. We are working on a novel treatment and a novel paradigm towards COPD. We think that COPD is basically a misunderstood disease. You could argue with it, is it actually a disease to start with, but we'll get to that in a few slides. The idea to this project comes from Iceland, of all places. There was the head of respiratory at the Iceland Landshospitalis, the major hospital, who was prescribing azithromycin, as you do for pneumonia patients. And what he realized was that if he prescribed this azithromycin, which is an antibiotic, to people who doesn't have an infection, but have COPD, cystic fibrosis, there are actually only one person on Iceland with cystic fibrosis, but there are a few asthma patients. They actually get a relief in their lung function. They start breathing better when they get azithromycin, even though they don't have a infection. So he was kind of looking into this and saying, okay, what's happening here? So he called up his friend at the BMC, the biomedical center, the Professor Thorin Gudjonsen, and asked if they can do some collaboration around this. And what they realized together was that azithromycin doesn't really have one mechanism of action; it actually has two mechanisms of action. One of them is it's an antibiotic. It is well known, well characterized, the mechanism of that. What they found out was that azithromycin also induces epithelial growth. So anything you have a breakage in an epithelium, if you treat them with azithromycin, the epithelium heals. And then they published a lot of papers around this in the early 2000s. I think around 2010, a med student came along listening to this story and raised his hand and said, "This is great, why don't you make a drug out of it?" And they didn't really know how to respond, and he's kind of impatient, so he started a company and said, we need to do something around this. And the basic idea, why we think that this is a misunderstanding, is a pointer here. This is how an epithelium should look like. If you imagine this is the healthy lung, you have a protection in both ways, so nothing from your body goes into the lung and the other way around. Now the problem is that with COPD, and that's why I'm saying maybe it's not a disease, it's not like you wake up one morning and you find out you have a mutation and all of a sudden you got COPD. COPD is more of a wear and tear disease of your lungs. So either you're a heavy smoker, or you live in London subway, or Beijing or something. What you will do is that you will expose your lungs to radicals, acid, bases, particles, anything. Over time, the lung can't really take this insult, abuse anymore, and what happens is it starts leaking. So you will get kind of a rupture in the lung. Now what happens when you get that rupture is that, of course, the body will start gathering neutrophils and granulocytes and send them out as a protection measurements. And what you will find then is an inflammation. But the inflammation is not a disease. The inflammation is caused by the rupture of the epithelium. And I think this is, to our thought, this is where everything kind of went wrong for the last 20 years or 30 or even more, is that COPD has been looked as an inflammatory disease. And it's very easy to do animal models for inflammation in the lungs. You can expose them to LPS or anything. If you give them an anti-inflammatory drug, you will see beautiful results preclinically, you will see beautiful results in the animals, but it's not translatable to the clinical trials. And they have tried this over and over again. And still they try. So we consider this rather a secondary effect. We have done our LPS challenge as well. We also show good results. We lower the inflammation. But we don't think that this is because we are anti-inflammatory. This is because we are strengthening the barriers, and therefore we are not allowing the inflammation to happen in the first place. Of course, if we are right, and we might be, there are of course different diseases that you can expand this to. Basically anywhere where you have an epithelium that is slightly leaking. You can think of gluten intolerance, leaky gut syndrome, ulcerous colitis, all the dermatitis regions. You have asthma, cystic fibrosis. All of those things are fair game if this actually holds true in the end. So what we have done chemically is that we have started with azithromycin. Azithromycin has been very well researched for 20 years by the major companies, Sempras, Glaxo, Pfizer. They have filed gazillions of patents around azithromycin as an antibiotic. Now, if you do some certain chemical modifications, you can remove the antibiotic feature of azithromycin. And that is what we have done. And then we added some other things on the side just because it's fun. I'm a chemist. I like those things. So what happens then is that as soon as you remove the antibiotic effect from azithromycin scaffold, the IP landscape becomes completely barren. It's a complete desert. So we filed a composition of matter patents in 2016. It's already proceeded to the intention to grant from the EPO authorities. And we are actually kind of actively delay it by adding silly claims that we are not allowed to add, just to push the approval. Because once it gets approved, we need to file in national phases, and that will cost us a lot of money that we don't have. So we know that we're going to get the patent, we just don't want it to be approved just yet, until we have the money. So the good thing about this, as I said, they are non-antibiotic. They retain the barrier enhancement effect. And we had to develop our own animal model, because as I said again, if we do this the ordinary way, we're only going to looking at inflammation. And we would like to look at the rupture and the breakage of the lung. So we are actually exposing the mice, in this case, to sulfur dioxide, which then they breathe it forms sulfuric acid inside the lungs, and of course that's kind of disruptive to a lung to have sulfuric dioxide. Don't breathe that if you can avoid it. If you work on Iceland, you will breathe it if you're working on the aluminum factories and the smelters. So they have some really nasty lung diseases over there. What we then do is that we inject human albumin in the tail vein, flush the lungs, and you can see that you, depending on how much human albumin goes out into the lung, that's a kind of a good measurement for how disruptive your lung are. And of course, we tested it. We have tried for different. This is just a summary. I think we have sacrificed 400 or 500 mice to date in this model. So this is just a brief section that we see that if we dose them 2, 3, 4 weeks in a dose-dependent manner, we look at the PBS, which is the untreated mice. We look at azithromycin treated and one of our compound. You can see that we can kind of protect up to 90% of the insult from sulfur dioxide by pre-treating them with our compound, which is really weird, but it works and it's good. So right now we are then looking for an investment to take this into clinical trials to see if it works also in humans. We're not going to expose humans to sulfur dioxide. That would be highly unethical. So we're instead using a mannitol challenge test. That's one. It has been developed mainly for gut diseases like IBD, I think it's called. So one way to check if you have a leakage in your gastrointestinal is that you eat mannitol and then you basically take blood samples. If you have mannitol in your blood, your gut is leaking. It's simple. So what we are doing is that we're going to have our SAD/MAD study as usual, the phase 1 in healthy volunteers, and then we're going to do a phase 1b study where we let COPD patients breathe mannitol. We treat them for 4 weeks and then let breathe mannitol again, and we check the mannitol level in the blood. If it goes down, we kind of have healed the lung. So it's not a full-blown phase 2 proof of concept, but it's at least something we can do to increase our certainty that we're on the right track in a phase 1 setting. And we're going to do this with David Singh, who is a professor in Manchester, who is one of the key opinion leaders in the field of COPD. So he's done this before, and he likes the concept and is now also a member of our scientific advisory board. So far we have tried to have this kind of semi-virtual. Of course, we need to do some research around it, both in vitro and in vivo, and that is done in Reykjavik under the supervision of Professor Thorarin. We have the professor who kind of prescribed this azithromycin in the beginning. Olafur Baldursson is now our CMO, and I didn't put him away. The chairman of the board is Professor Clive Page from King's College here in London. Myself, being the CEO, situated in Stockholm, Sweden. Our toxicologist is also in Sweden. Our regulatory consultants most likely going to be in Netherlands. So we are a European kind of virtual company. So far, all our work has also been done with reputable companies that you probably know all about them. So what we are now trying to do is raise 6 million euros to take us through the phase 1 and show well, it's not really proof of concept, right? But at least we should show that we can lower the mannitol level in the blood for patients. And I think in the interest of time, I can stop there. Right.